Welcome, everybody. My name is David Pellman, and it's a real pleasure to introduce Yukiko Yamashita for our Connect Science series. Yukiko is a new member of the Boston biomedical research community, and it's great to have the opportunity to introduce her, at least to the Dana Farber group here. I've been following Yukiko's work for many years. She's done really remarkable work on asymmetric cell divisions, the maintenance of stem cells, and germ cell immortality, the maintenance of rDNAs, and most recently, near and dear to my heart, she's also been working on how nuclear bodies called chromobodies are organizing chromosome in the nucleus and helping ensure the formation of a single nucleus after cell division. Yukiko is originally from Japan, and she did her undergraduate and PhD work at Kyoto University with in a very famous lab, Mitsuhiro Yamagita's lab, one of the main early labs working on cell division in yeast. She then moved to Stanford and did a postdoctoral fellowships with Minx Fuller, where she developed her interest in stem cells and asymmetric cell division. She took her first faculty job at the University of Michigan, rose to the rank of full professor with an endowed chair. And then in 2002, the Whitehead Institute was lucky enough to be able to lure her over, and she is currently a professor in the Department of Biology at MIT and the Susan Lindquist Chair at the Whitehead Institute. Yukiko has too many awards to go through for time, but I will highlight that she is a MacArthur Award winner and an investigator of the Howard Hughes Medical Institute. Now, after that quick walkthrough of her CV, you'll be very surprised to know that Yukiko, in our discussion just before the introductions here, claimed to me that she's a very lazy person. I find this hard to believe, but she does claim to be a lazy person who, if she hadn't been a scientist, would have been a dog walker. I don't know if that'll be evident in the talk, but I'm certainly looking forward to hearing from some of the unlazy side of Yukiko's endeavors. Thanks so much, Yukiko, for joining us.
Thank you, David. So I'm really excited to be here to give a talk today. I think probably my laziness kind of show in my science. I mean, my fascination in science, but at the same time, I always feel like I was dragged into the new field. Often case, the data nags us like, you know, you got to be studying this. And I just, you know, of course, I'm excited to do science, but always, you know, going into the new field, typically, you know, the nagging evidence really tells us you got to be studying it now. So with that, you know, today's, you know, the topic I'm, you know, telling you about, that's something we are very interested in now. So, yeah. But at the same time, you know, that we really unexpectedly get into this field. So but before going into that, I want to just pick you with one question. So we all know that our somatic cells have only a finite number of the time they can divide before they go aging or senescence or death. But if you really think about this fact, our germline lineage, right? Because all of us came from a germ cell or two germ cells from our parents. And then that also, you know, who used to be also a single cell that came from each, you know, the two germ cells from their parents, which is our grandparents and so on. Meaning that, so this germline lineage lasted for the evolutionary time scale And if you think about even just a multicellular organism's history, that's 1.5 billion years. So germline lineage has lasted that long. What really make it possible is, I think, actually a really fascinating question. But of course, we didn't start our work, the really trying to address, okay, we're going to study germline immortality, which felt like just way too big, to start tackling from any other aspect of any of the aspect. So we, our long-standing, the interest was in the asymmetric cell division, which is a process to generate two cells with distinct fates, meaning starting from one cell and then you end up with two daughter cells that has a different fate from each other. So the original reason I got really interested in this was, I worked my graduate, throughout my graduate career, trying to understand how cells make exact copy of each other. To make sure all of those DNA is the same, contents are the same, and after the cell division, you have two exact same cells. So that means to me, really, the thinking about the asymmetric cell division is really fascinating things because, they duplicated pretty much everything equally, but they, go to take on a different fate. And this asymmetric cell division is a really important process because, that we, as we all know, that we started our life as a single cell, as a zygote. And then this zygote had to go through many rounds of asymmetric cell division. Otherwise, we cannot be this, the human being, the multicellular organisms having almost 100 trillion cells in our body. The many of cells having very distinct function to make us as an individual. And also asymmetric cell division plays an important role during the, the tissue homeostasis. One of the prominent example being stem cells asymmetric cell division that creates one stem cell and one differentiating cells upon division to make sure that, the tissues do not have too many stem cells or too many differentiated cells. Okay, so we study this process using Drosophila male germline stem cells as a model system. So out of this, the male flies, tiny, Drosophila, you can take out the testes in a matter of 10 seconds or so. And you can, the beauty of tissue is you can see all stages of differentiation from the stem cell to fully differentiated spermatid in one tissue. So this is, the tissue of one snapshot. You can take a look at all, the stages of the differentiation. And then what we focus in particular, especially for this talk, is around here at the very apical tip, which is a blind end of this, the tubular testicular structure where you can find stem cells. So the beauty of this tissue is that you can identify all cells at the single cell resolution. So by the combination of multiple cellular markers and etc., you can identify the germline stem cell. And then these orange cells now, that I'm showing as a, just a circle, the germline stem cell, you can identify them at the single cell resolution with almost 100% precision based on their anatomical location. And then what is the anatomical landmark, which is, shown here is a cluster of green cells over here, which is called hub cells. You don't have to remember this name, but this functions as a stem cell niche, meaning these cells are equipped to send out the signaling ligand, the very short range signaling ligand to the neighboring cells, which is a stem cell, to specify or to instruct the stem cell program. So that's why, the receiving the ligand from these cells, the stem cell can specify their identity as a stem cell. And whenever these stem cells divide, this division will create always one differentiating cell. So this is always stereotypical asymmetric stem cell division. And not only that, we can identify all these cells at the single cell resolution. We could even identify stem cell and the differentiating daughter pair that came from single stem cell division. So this ability and precision gives us a lot of power for us to study what's going on with the asymmetric stem cell division. Okay, so so today I'm going to tell you about a little bit of unusual way of thinking about the DNA, the chromosome segregation. So as I said, we tend to be dragged into a field, and we are also dragged into this field to think about the non-random sister chromatid segregation. And then this was quite unwittingly because we didn't believe about two sister chromatids being different. And we felt that that's a violation of the idea of the DNA replication. Whole point of DNA replication is it to be very, very precise and to make exact copy of each other. So as you can see here, here's a DNA strand, the double helix, right? Plus strand and minus strand. During DNA replication, these plus strand and minus strand would split from each other, as you all know, to template newly synthesized strand, right? So being a very, very precise procedure of the DNA replication, these two pairs of the DNA strand, double helix, is called a sister chromatid and supposedly identical. However, so there was some, the ideas or hypotheses had been put forward to say that maybe one of the sister chromatid may be associated with some sort of distinct information, such as epigenetic marks, or sometimes, perhaps a mutation loss, because DNA replication introduce some mutations, right? Or whatever it is. And if those distinct information is segregated to the one daughter of the cell division during cell division, maybe this kind of not necessarily confers, but maybe at least underlies to the asymmetric outcome of the fates. So that's been the idea. And it's been highly controversial because in many cases, experiments to try to address this possibility was not quite, how to say, the sufficiently high resolution to be able to give you a very confident answer whether or not this, the chromosome segregation was indeed asymmetric or not. So, so with this background, my former student, currently actually assistant professor at the University of Michigan, so Swati Yadrapari, wanted to ask this question with the highest possible, resolution/rigor. So she came up with a very genius way of addressing this question, combining the method called chromosome orientation fish, co-fish, which this protocol itself was out there, but she added some really smart, the twist to this technique. So in this protocol, what you do is when cells are undergoing, the DNA replication, you parse them with BRDU so that only newly synthesized strand would contain BRDU. And then, so BRDU containing DNA strand is 500 times more sensitive to certain wavelengths of the UV. So you can nick them, and then, after the fixation, you can nick them, and then you can apply some, exonuclease to remove those strands, leaving only template strand as, intact and single stranded. By using this plus strand from minus strand difference, they are not the same, just they are complementary. By using this, you can apply strand-specific probe to differentiate which sister chromatid had which strand as a template. And then, so that this method called co-fish itself has been, out there for quite some time, by the time Swati applied this, protocol. But, the one really great idea that she applied was that she realized she could design a chromosome specific probe. Some, the chromosome X specific probe, chromosome 2 specific probe, and so on. So in this manner, Now she could, ask which strand of the sister chromatid of which chromosome went to which side of the asymmetric cell division during stem cell division, meaning maybe red strand goes to the stem cell, then opposite strand will go to the differentiating cell. So this is an example, exactly how it looks like. And as I told you, we can identify stem cell and the differentiating cell pair that came from the single division at the single cell resolution. So then if stem cell gets a red strand, in this particular case, this is a Y chromosome probe. If stem cell gets a red probe, differentiating cell will get the blue probe. And this complementarity, instead of stem cell and the differentiating cell both getting red or both getting blue, that would tell us, the experiment didn't work. But because we always see red and blue or opposite situation, blue and red, we can tell this is actually, the very good sign that the experiment worked out. Okay, so now if the two sister chromatid that came from the DNA replication process, if they are really identical to each other, it shouldn't matter whether stem cell gets red strand or blue strand, right? So in theory, it should be random, meaning these two patterns of chromosome segregation should be observed 50-50 of the chance. However, when Suwasi looked at the X chromosome of the two sister chromatid of X chromosome, I'm not talking about the homologue like X and Y. So two sister chromatid of X or two sister chromatid of Y, she saw a striking bias where so-called red strand going to the stem cell side with really, high frequency, like 80% or more than 80% of the time. At the beginning, we worried that this got to be something, because of some sort of experimental artifact. However, if you try the exact same experiment with the autosome probes, now we don't see that bias, telling us that probably this is actual biological phenomena instead of some sort of experimental artifact. And then at that point, we are very fascinated by this phenomena. However, we had no idea how this happens or why this happens. So that was 2013 for us. And around that time, the Suwasi graduated and the new postdoc, George Watase, came in, and we sat down together and then talked. We know it's going to be very difficult, but if we want to really, make any progress, on this front, we have to map the chromosomal element that is required for this non-random sister chromatid segregation. And then we postulated at the least there should be a the chromosome specific cis element, because second chromosome, third chromosome do not behave the way X and Y did. So cutting very, very long story short, because it's already 8 years from 2013, cutting a very long story short, George realized when we use a chromosome that deletes a huge region of, X chromosome heterochromatin, now that X chromosome was segregated randomly. And what's in this region? And we figured out there's one is a ribosomal DNA locus, and the other was, the 359 base pair satellite DNA repeats. And if anybody asked me, which is your favorite candidate, I with no doubt, I would have said the satellite DNA. Our DNA got to be boring. That would have been my, the initial, the reaction. And then as a matter of fact, that sparked a completely different direction of the research in my lab studying satellite DNA. If anybody wondered, if anybody knew that we are working on a satellite DNA as well, and then why we started working on it, that was because of sort of, the misperception at this point. Okay, but then, so further mapping this, we turn to the Y chromosome. Y chromosome also contains this ribosomal DNA locus. And then there's a, the nice chromosome that specifically deletes ribosomal DNA from Y chromosome. When we looked at such a chromosome. Such Y chromosome also segregated randomly instead of non-random. That gave us kind of a surprising answer that ribosomal DNA is likely responsible for non-random sister chromatid segregation. So we didn't necessarily like this data because we didn't know what to think of this, but we had to admit that kind of made sense. Because two autosomes, second chromosome, third chromosome, that we knew did not segregate non-randomly, right? They do not have ribosomal DNA locus. And moreover, more recently, the George looked at the translocation chromosome where second chromosome acquired ribosomal DNA from the Y chromosome by translocation. This chromosome now segregates non-randomly, telling us probably ribosomal DNA locus is sufficient to do this. And then, again, I'm cutting a little bit of, long story short, but, within the ribosomal DNA locus, we had a reasonable guess to think that intragenic spacer, which is a portion of the ribosomal DNA that does not generate the ribosomal subunit, is likely being required or important for the non-random segregation. So based on this idea, we decided to now isolate the trans factor, the protein factor that binds to ribosomal DNA, IGS, to perhaps the mediate non-random sister chromatid segregation. So we the synthesized the IGS, put onto the, the strip, the avidin beads, and then, the sprinkled the germline stem cell extract, and they did the mass spec to identify some candidates. We conducted secondary screening based on the localization to the ribosomal DNA locus. If you take a look at the chromosome squash, this, the new protein that we isolated, which we named Indra, localized to ribosomal DNA, and we can also confirm it's highly, enriched on the IGS sequence specifically by qPCR. So we identified some other proteins, but for the sake of today's talk, we are focusing on this new protein, Indra. This is a novel zinc finger protein. And then obviously now that we have the candidate protein factor, we had to ask, is this required for the non-random sister chromatid segregation? And yes, so compared to the control situation where you observe this bias, knocking down the Indra, that caused complete randomization of sister chromatid segregation pattern for the X chromosome as well as Y chromosome. So at this point, as you might be aware, or you might realize, we were in a kind of a funny situation. At this point, we knew the molecular mechanisms, at least the molecular player of biological phenomena whose significance remains unknown. So what's the point of having a molecular mechanism of something that we don't even know the meaning, right? And also, even if we know it's rDNA, and also, we have, the proteins, etc., we still didn't know what is the, the difference between a sister chromatid of ribosomal DNA locus. Okay, so to answer that question, I have to step back a little bit to explain what the ribosomal DNA looks like. So if you look at your cells, like 60% of all RNA transcription is coming from transcription of ribosomal DNA to generate ribosomal RNA to make ribosome. And because of this high demand, ribosome genes are repeated many times on our chromosome, like, hundreds to thousands of times, on our genome, making up so-called rDNA locus. In case of Drosophila, as I already, told you, rDNA locus happen to be on the X and Y chromosome in Drosophila melanogaster. In human, they are on autosomes, and I think there's a 5 loci, if I'm not mistaken, per haploid genome. It's around that number. Okay, so the problem of having this, the highly repetitive tandem repeat locus is that although it's important to keep up with the demand of ribosome biogenesis, this, the tandem repeats often result in intrachromatid homologous recombination because there's so many homologies along this, the locus, right? So that end up with leading to the copy number loss in the form of this, the rDNA circle, reducing the copy number on the main chromosome. And this instability of ribosomal DNA is known to be a cause of the senescence in the yeast, okay, in the, unicellular organisms. However, not much has been studied about this, the impact of losing copy number from the rDNA locus in the multicellular organisms. So, my former student, Kevin Liu, wanted to study this aspect. And this is what we found, squishing his 4 years into, again, one slide. So here I'm plotting the copy number on the y-axis and then time or generation on the x-axis. And, if Drosophila has over 130 copies of the ribosomal DNA, they are happy, which is shown in this green range. Below that, first you go into still alive, but you start having some sort of a phenotype. That's an insufficient phase. Below 90 copy numbers, now they are dead. Okay, so if you look at any flies, wild type flies, typically those flies have above 130, this is in the green range, right? But when Kevin looked at, the copy number during aging in a male germline, okay, so he was able to detect a copy number loss. So as these male flies age, his testes start losing the copy number of the ribosomal DNA. And then of course, the germline, makes sperm ultimately to pass on to the next generation, right? And sure enough, so he found that the new progeny, young progeny coming from older father actually have inherited reduced rDNA copy number. I mean, this is, of course, the ones you are told, everybody would think, yeah, this is obvious, right? Because, the DNA comes from the parents and that should be, of course, heritable. But then, now you realize this is a big problem because if you keep losing rDNA copy number, soon enough, in a few generations, you're going to go into this domain of the insufficient copy number, and then you're going to go extinct. But clearly, none of the flies appears to have gone to extinction, and neither do we, right? So surprisingly, these young flies who had to start their life with a low, the lower decreased rDNA copy number, his testes now can increase the copy number so that F2 next generation actually had, the recovered copy number. So when we saw this phenomena, we realized something similar was described half centuries ago, which was called rDNA magnification. And back then, people were studying this, the really weird chromosome or mutant chromosome that had significantly / drastically reduced ribosomal DNA copy number. And that mutant chromosome is down here, insufficient copy number showing exhibiting some sort of, the cellular phenotype and then, the developmental phenotype. But you could induce those chromosomes to recover the copy number. And then in one generation, you could induce to have those chromosomes recovered to the sufficient copy number range. And this process has been called rDNA magnification. And then we realized, oh, this is probably the same thing. And indeed, we found exact same gene sets are involved in these two phenomena, rDNA magnification as well as copy number recovery from the reduced copy number due to aging of the parents. Okay, so that means, 50, 40 years ago, the meaning of rDNA magnification was kind of unclear. But now we think that's a manifestation of the regular maintenance mechanism that normally happens as a fluctuation in a wild population. Now, if this is the magnification, now there's quite a bit of a good hypothesis or molecular mechanism about how magnification might happen. So this was what we figured out about already 50 years ago, that this hypothesis was put forward based on quite convincing, the evidence. So here I'm showing just, 8 repeats, but this is ribosomal DNA repeats. And then, here, the red and the blue, this is a sister chromatid of ribosomal DNA. Okay, if this copy number is reduced to certain, the extent, and if you want to recover, how would you do that? So what they do is they misalign these sister chromatid on purpose, right? And then they find the homology with each other on the misaligned location. And then you undergo exchange, and this is called unequal sister chromatid exchange because this results in unequal sister chromatid eventually, meaning one sister chromatid actually stole the copy number from the other sister chromatid. And when we drew this, this diagram based on what was proposed 50 years ago, we were completely shocked. And then we realized, oh my God, this is an asymmetry we have been probably looking for, right? So what is the basis of asymmetry that let the cells differentiate to sister chromatid, leading to non-random sister chromatid segregation? We thought maybe this is a copy number. Okay, so that became our idea. When stem cells are dividing to make a differentiating cell, can we see ribosomal DNA copy number asymmetry, like, to see the recovery process, right? So then, that was probably already a few years ago. We got really excited, and then George did the DNA in situ to quantify, rDNA copy number when stem cells are undergoing mitosis, when cells are in anaphase, so that we can see stem cell side and then, the differentiating side. And maybe, as you can appreciate here, or maybe you cannot appreciate here, actually there's no copy number difference. I mean, we couldn't just see any convincing, the asymmetry, because sometimes, the one side looks more than the other, but that could be anyway. So we got very, very disappointed. And then we have been talking for a while, maybe, the copy number recovery is under the detection limit, right? Because, out of 500 copies, if there's, that becomes 10% different between these two sides, maybe we can't differentiate. So we were pretty disappointed. But then one day, George came to me like, maybe we have been looking at the, the wrong condition, wrong genetic background, because what we are looking at was actually wild type, so-called non-magnifying condition that contains sufficient ribosomal DNA copy number. Oh, so if that's the case, we maybe have to induce this copy number recovery by giving them, the magnifying condition, meaning X chromosome have bare minimal ribosomal DNA, Y chromosome have no ribosomal DNA. That's this situation. Y chromosome have no rDNA, and X chromosome have a little bit of rDNA left. As soon as we did that, we started seeing the asymmetry, and a more copy number was going to the germline stem cell side. And then that is about 20% more or 20-30% more. I don't think we would have believed just this data because that's, being 20%, right? Although we are very careful, still 20% can be, I mean, we don't know. I don't know if we were convinced enough. But after many years of trying, I mean, not really trying, but, wanting to see the asymmetry in a wild type situation and then not seeing it. And in that kind of wild type situation, there's absolutely no asymmetry, as you can see over here, right? It's completely 50-50. After seeing that, we are very, very sure it's interesting, it's happening, okay? And then on top of that, if you mutate INDRA, this is a protein that binds to ribosomal DNA and then required for the non-random sister chromatid segregation. This goes back to, 50-50. So this striking difference now, really convinced us this asymmetry is, like really happening. That's a real phenomena. So we wanted to have a little bit of more concrete, the evidence for it, because, there's still, this is a kind of, unconventional idea, and just relying on one data point or one kind of data, that we don't have, of course, the enough courage to say, yeah, this is what's happening, even though it makes sense. So, okay, this is, again, the diagram of sister chromatid, the unequal sister chromatid recombination. And then, of course, the idea is this is asymmetry, and then that I showed you in the previous slides. Another thing of unequal sister chromatid exchange is that, exchange itself. That means, right, the blue strand and the green strand and the pale green strand, you can, if you exchange, you do see the flipping of the, the dark green versus light green. So, and then when I said we were doing the co-fish, we are using this, the 359 probe as a, to assess sister chromatid segregation pattern. So when we say X chromosome's red strand is going to stem cell side, it was actually red being 359, the location over here, okay? And then if you just take a look at the, the wild type situation, non-magnifying condition, when cells have enough rDNA copy number, you could use other side of the ribosomal DNA. This is TAGA satellite DNA. And you do the exact same, the co-fish procedure, we still see red strand going to the stem cell side. So either TAGA or 359, both case, red strand goes to the stem cell side much more frequently. Now imagine if exchange happens, right? So the light green and the dark green would flip. Then, so the 359 and TAGAs, red versus green, red versus blue are going to flip. Instead of both red facing to the stem cell side, one might face to the stem cell side, but opposite side, the opposite strand, blue, is going to face to the stem cell side, right? And then exactly that's what we see. So now 359 actually remains to be segregated to the red side, but then, the TAGA side now start flipping, and then now start giving blue strand to the stem cell side. Okay, so that gave us a kind of confidence. I think, that we could probably say ribosomal DNA undergo sister chromatid exchange during magnification, and then start giving TAGA side, the blue strand to the stem cell. And then not only, so this data, number one, shows there is an exchange, right? But a second thing that strikes me is that, before going that, one thing that strikes me is that for some reason, this side, right, of the chromosome, the 359 side is fixed to go to the stem cell somehow. I don't know why. It really looks like this side might have some sort of information to keep certain chromatid to the stem cell side. And with that said, it's interesting in a normal situation, without any need of increasing copy number, therefore, in the absence of copy number asymmetry, still stem cell knows to segregate this red side to the stem cell. So I can discuss a little more about this implication, but I think I found it to be very fascinating. Okay, so then, the remaining point on this discovery is that is INDRA required for ribosomal DNA maintenance, right? Because, if all our idea is correct, the INDRA should be required for ribosomal DNA maintenance. Sure enough, if you make the INDRA mutation, first of all, transgenerationally, fly lose the fecundity. So compared to the wild type, which is, this gray, gray points, they of course maintain the fecundity across many generations. Otherwise, they go extinct. But upon INDRA knockdown, you can see that the fecundity decrease dramatically, across the generations, and which is also associated with a ribosomal DNA copy number decrease along the course. And interestingly, I want to point out that you can see over here, right? It kind of decrease in the RDNA copy number plateaus around here. And we think this is because they are hitting the really rock bottom. Below certain range, they don't even survive. So we can't even, see them or, as an animal to be able to actually do the this digital droplet digital PCR to quantify ribosomal DNA copy number. Okay, to summarize, so what I told you is this is really unexpected mechanism and the meaning of non-random sister chromatid segregation. As opposed to previously, the proposed hypothesis that non-random sister chromatid segregation comes from across the chromosome, some sort of epigenetic or some sort of information from tail to tail of the chromosome. Instead, this information is localized to ribosomal DNA. We think this asymmetry is RDNA copy number difference. That's the asymmetry. And we also identified this novel RDNA binding protein INDRA to mediate this process. And all these things is for the sake of recovering ribosomal DNA copy number. Okay, and then, so then this really contributes to the germline immortality. And that's how, we came to realize, okay, we are studying the germline immortality. And as you can tell, right, because, if you remove the INDRA, you end up with losing all those, the fecundity and then ribosome, together with ribosomal DNA copy number. By the way, why did we name this gene INDRA? This is after his god, Indra, who, being God, he was immortal, but, because of the curse, he, fallen mortal because of the curse. So, based on, the tradition of the Drosophila genetics, you have to name the gene based on their mutant phenotype. We thought this, the name Indra was very fitting. And then in the last just a few minutes, I'm gonna spend a little bit of, the additional project in the lab that also matched down to the ribosomal DNA. So I told you that, the sister chromatid exchange has to happen to recover ribosomal DNA copy number. First step to it is double-strand break. Who is making this double-strand break was a kind of interesting question. And then, so we actually didn't really think that this was what we are studying when we started this, line of work. Around 2016, the Jonathan Nelson joined as a postdoc in the lab. He was interested in this weird transposon that specifically insert into ribosomal DNA locus called R2. This is non-LTR retrotransposons. And a really weird thing about this transposon is that it's really sequence specific and only goes into ribosomal DNA. And if transposons are the genetic parasites as we normally think, right? How come we host let transposons goes to the most important part of our genome? So we thought it didn't quite make sense. And then we, the John wanted to study the function of the R2 retrotransposons. So then, I probably don't want to go through all those data, but, what John found is that R2 is required for stem cell maintenance, germline stem cell maintenance. If you knock down R2, and then you can see that the germline stem cell quickly, the decrease compared to the wild type control. So suggesting R2 is somewhat functional, to the host. And then somehow, it's beneficial, right? And then this benefit related to the ribosomal DNA because they insert into rDNA. And indeed, it seems to be the case. First of all, when they are losing the germline stem cell number, upon knockdown of R2, so we also detect the ribosomal DNA copy number decrease. And then germline stem cell loss can be rescued if you supplement the mini X chromosome that contains extra ribosomal DNA copies. And then, so I think I'm going to probably skip through because that is quite a bit of, the genetics. But what I wanted to show here is IL-2 is actually required for the process of rDNA magnification. So I'm not going to show go through the actual data, but the bottom line is in the absence of IL-2, flies lose the ability to recover rDNA copy number. Okay, and okay, so then another striking thing is, first of all, we found that when rDNA magnification is induced by having fewer rDNA copy number, you can detect DNA damage, which is, the gamma H2AX, phosphor H2AX staining, shown in red over here. Stem cell have this DNA damage, okay? And that we think now is the, the sign of the, fast double-strand break to induce ribosomal DNA copy number recovery. If you do the RNAi, now we stop seeing that kind of, the ribosomal, the DNA damage in the stem cell. So this is a graph. If you induce the magnification, you induce the DNA damage in the stem cell, which is completely suppressed if you also knock down R2. And then one thing, probably you might wonder, R2 is a retrotransposon. That's the parasite. How the benefit and the harm can be, the balanced, in between. And what we think is now, the R2 is normally silent, perhaps, based on the pyrRNA mechanism, that kind of defense mechanism of the host. In a normal situation, the label is actually the flipped, but in a wild type situation, when they have enough copy number, we don't see R2 expressed at all. But once copy number goes low, we start seeing the expression of R2. So this is what we see, under the magnifying condition, R2 started waking up. So R2 is induced only when it's necessary. And, that's how, okay. And then the final question we wanted to ask is R2 is required for this germline immortality. Indeed, it looks like the case. If you do the R2 knockdown, then, this transgenerational fecundity or, the viability or, the continuation of the strain is completely compromised compared to the wild type that can, of course, pass to the sixth generation or, many million years they have been passaged, right? Compared to the wild type, R2 knockdown leads to quick, the really rapid extinction of the lineage. And that is also associated with a decrease in the rDNA copy number. So that said, R2 is required for the germline immortality through rDNA maintenance. And this is, I would say, this is a very unexpected function of retrotransposon. And to summarize, so we really bumped into this, and the ribosomal DNA copy number is really critical aspect of our genome. During aging, that can get decreased. You have to recover. To recover, you have to make a double-strand break, which is now we think is mediated by R2 retrotransposon. And after that, the sister chromatid undergo unequal chromatid exchange, which leads to non-random segregation of the recovered copy number to the stem cell, so that the recovered stem cell can generate many more the progeny, which becomes eventually sperm, to propagate this recovered copy number to the offsprings. So the recovered copy number seems to be going to the stem cell, and this process seems to be mediated by this rDNA binding protein INDRA. So with that, thank you so much. And the part of the INDRA, non-random sister chromatid segregation, has been really single-handedly carried out by George Watase. And the R2 part was carried out by another postdoc, John Nelson. And thank you very much. And I'm happy to take any questions you might have. Thank you for your attention.
I'll clap for everybody. Thank you, Yukiko, for a really thought-provoking talk. That was great. Just for the audience members, if you can just make sure to post your questions in the Q&A and not the chat. I'll try to read some of them. And here we go. This is from Jonathan Frieden. Does non-random sister chromatin segregation confer some evolutionary advantage in fitness? If so, by what mechanism?
So that's really interesting.
Chromosomes?
Yeah. So what, this cell type we're looking at is still undergoing pretty much mitosis and not meiosis. So sister chromatin being probably, other information should be equal. So we don't necessarily think that's really, related to something like meiotic drive. But of course, that kind of evolution, I don't know if it's going to drive it or not. But with that said, when these cells eventually undergo meiosis, it's really funny. In this species, ribosomal DNA also functions as a pairing site between X and Y chromosome. So I don't know. I don't know what to make of it yet. I don't want to make, the overcommitting, the stupid thoughts over here. But I wouldn't be too surprised if it also contributes some sort of, the evolutionary selection process. And of course, in terms of just a copy number, of course, you can't lose the copy number below certain number. So in that kind of negative selection, of course, the rDNA maintenance is, of course, under the selection. But I just assumed that you were asking about the positive selection. That I don't know. I don't have a clean answer to it. Yeah.
Great. Sarah Pierce wants to know, can you please comment on whether human cells, I'm sorry, this is jumping around, exhibit similar non-random segregation of the autosomes that contain our DNA?
Yeah, we'd love to study that. However, well, we want to really see that, but in human or even mouse, we don't have a cell type that consistently exhibit asymmetric cell division, right? We cannot, there's a, the cell type that is postulated to divide asymmetrically, once in a while, based on all those modeling and etc. But, we can't say, oh, this cell just divided asymmetrically. We can't identify that kind of cell type. Nobody knows which cells actually do this. So with that said, we can't do that level of analysis, like looking at the red strand and the blue strand that went to this side or that side, because which side, there's no landmark in, the mouse or human, the cell type that we can tell, oh, this is one particular side, not just the asymmetry in a copy number. So with that said, I think eventually we can at least, what we can check is, for example, if we can identify the ortholog of Indra. But by the way, it's zinc finger protein. So, if you are just talking about the homolog, it's just a ton of it. But if we can identify true ortholog of Indra in human cell, mouse cells, we should be able to do the similar experiment, at least to see if that's required for example, transgenerational maintenance of ribosomal DNA copy number. That's what I would go after, to address your question. But yeah, we can't actually test this specific asymmetry, at least with the current knowledge of, human and then mouse cell types.
Great. Kai Wucherfennig wants to know if Indra or R2, or I suppose their orthologs, are expressed in cancer cells, and how could they contribute to
yes. Yeah. I want to draw the parallel between rDNA copy number maintenance and the telomere copy number maintenance. Both of them are, the constitutive the requirement for the chromosome, right? And then also, in case of rDNA, its actual enzymatic function as a result of, the transcribing rDNA is, essential for, the cell survival. Both telomere and rDNA have an exact same kind of requirement to maintain a copy number, although, although the cellular activity is constantly degenerating that copy number, right? Always, every time you divide, you are losing copy number. I mean, maybe for the different mechanism, right? Intra-sister chromatid, the recombination versus, the endo-replication problem. But same idea, both of them keep losing the copy number. How do you maintain them? And that's really critical for any cell if they wanna, stay immortal. And the same thing applies to the cancer cells. And then, as telomere is somehow, the elongated, at least the telomere is a requirement to keep the cancer cells immortal. So with that said, I would really predict, the rDNA copy number maintenance should be also activated in the cancer cells. Yeah. And if they are expressed in the cancer cells, we don't know. And I think we seriously should look into, the true intra-ortholog in the mammalian cells.
Okay. From far away, we have a question from Peter Lansdorf, who says this is a fascinating talk and wants to know, how do the stem cells reproducibly end up with the elongated rDNA tracts? Or is the process random and are elongated rDNA tracts selected?
So, so that's something that I have mentioned in a talk. So we don't think cells are capable of counting the copy number, right? This strand 1, 2, 3, 4, 5, 6, 529, and then the other strand 1, 2, 3, 4, 5, it's 327. Oh, that's why the stem cell gets this. It's first of all, it's unlikely, and the second of all, as I said, in the absence of sister chromatid exchange, which means two sister chromatid has the exact same copy number, still we saw red strand went to stem cell. So that means we would like to say the propensity of copy number increase is inherited by the stem cell. How come you can do it? So we think it's probably some sort of biochemical difference between two sister chromatid. And if you think about the knowledge from yeast, so the ribosomal DNA is replicated unidirectionally because of so-called replication fork block. So that means, after the replication bubble opens a little bit, right? And then typically replication fork goes the both way in the most of the genome. But at the ribosomal DNA, right after the replication origin, there's a fork block on one end. That means DNA replication can go to only one direction. That means, on one strand, most of the region is replicated as a leading strand. The other strand is replicated mostly as a lagging strand. I think this difference can serve as a basis of the biochemical difference between two sister chromatid, to which, let's say, specific protein can bind, that binds to, let's say, Okazaki fragment and then mark that strand, that kind of things. And then in case of yeast, I don't remember which is which, but I think when DNA break happens to induce the sister chromatid exchange, that was, I believe, on one side specifically. I think I don't remember leading or lagging. Specifically, one strand breaks. Based on which whether it was leading or lagging. Let's say that was always the leading strand that breaks. So then that creates, and then you can actually take a look at the geometry. If you break at the edge of the leading strand, there's not much of the substrate for it to recombine to undergo unequal sister chromatid exchange, because you cannot go over beyond the replication fork because it has not replicated. You have to go backwards. If you go backwards, it's that strand that's going to increase the copy number. So I think the leading lagging difference can be tightly linked, the geometrically, or, to the likelihood of that strand gaining copy number versus losing copy number. So I think that's what the cells are utilizing. But it's sorry, after a long answer, this is a pure speculation. Yeah, thank you for asking that question.
Okay, actually, I have a question about, can you really rule out a counting mechanism? I mean, what if?
Oh, no, I don't. Yeah.
Indra is kind of like a counting.
Yes, you could, right?
And
yeah.
if Indra bound a microtubule motor, couldn't that just create an asymmetry that would drive segregation?
I think that's something, we are also thinking. But it's not pure counting mechanism for two reasons. One, the reason I already told you. Second, the reason that I'm going to tell you. First is that without the sister chromatid exchange, we still do see the non-random segregation. So that seems to tell us that the counting mechanism cannot be the whole thing. But with that said, if you look at actually in the amount on the segregating chromosome, okay, this blows my mind because it doesn't make any sense, but that means if we can figure that out, that's going to be probably the answer. Okay, without any exchange, in the wild type situation, we do see more indra on the stem cell side, and then, the less indra on the differentiating side, okay? So there's an asymmetry. In the absence of a copy number asymmetry, and as soon as we induce the copy, the sister chromatid exchange by having fewer copy number, now you see more rDNA on this side, fewer rDNA on this side. But then at this point, indra's amount is exactly the same between two sister chromatids. If anybody can tell me what this means, I would really appreciate. There should be some sort of really smart mathematics that might really make whole sense. Yeah.
Okay. We're running out of time, and I'm just going to have one more question, and that's from Emma Sadir, who's asking, do stem cells consistently inherit the nascent rDNA transcriptional sense strand or the antisense?
Oh, yeah. So that is also, which is a sense, which is an antisense. That's also completely tied with the, whether it being replicated as a lagging or leading strand. We are trying to test that idea. We are trying to do the, the temporal inactivation of the ligase, DNA ligase, so that Okazaki fragment is not processed, okay? Or they are not tied together. All of those fragments are not ligated. And we are trying to use that to digest all newly synthesized strand to see if whether lagging or leading that's consistently going to the stem cell side. That's gonna answer your question. We don't have the answer yet. Thank you.
Okay. There are other questions, and hopefully Kiko might have time to take a look at them and send some responses, but let's just all virtually applaud and thank you, Kiko, once again.
Thank you so much.